Good morning, everyone. It's nice to see you back. And we are looking forward to some very exciting talks today. We'll start with Dr. Raghunathan from NCL Pune. So one of our research interests is to do constraints-based flux balance modeling. And today we'll get to hear from her about the constraint-based reconstruction analysis of metabolism using genomics data. So we'll just start the session.
Okay, thank you for introducing me. And at the start, I'd like to thank Arijit for the wonderful hospitality and also setting up this very focused, stimulating meeting. And all of you who have come down here early on a Saturday morning and have been waiting for almost 40 minutes quite patiently to hear me speak. So the title of my talk, as seen here, is From Genomes to Life, Constraints-Based Reconstruction and Analysis of Metabolism. So herein I haven't put any test system that I work on, and that was because I wasn't quite sure initially what the theme of this meeting would be, and I wanted to keep it very open-handed. And when I talked to Arijit, he told me, Anu, just talk about what you're most passionate about. And I realized that's a harder choice because I'm quite passionate about, or for people who know me, I'm quite passionate about almost everything. So I've decided that today's talk, I will give a little bit of background, some legacy data. I'll talk about one of the difficult diseases in the world, that's cancer, difficult to cure, difficult to understand, difficult to study. And I'll also give a short problem description of what we are doing and what is our model system, because every problem needs a model system. I will, for the benefit of students and people who are unfamiliar with the constraints-based philosophy, give a briefing about the methodology, and essentially within that methodology talk about some of the basic principles of reconstruction, what is the stoichiometric matrix, and constraints-based modeling, and then move on to discuss some of our research in the space of brain cancer, looking at glioblastoma, with specific reference to metabolic reprogramming. And that's illustrated better in this slide, where we use a systems approach, a systems biology approach, where we combine technology, computation, and biology to get insights into the disease mechanisms, get insights into why certain cells get resistant to chemo drugs, and how can you then tap into that understanding so that you can either resensitize or find alternate medicines for cancer. So some of the technologies we use are exome sequencing, getting transcript abundance, metabolite profiling, both exometabolome data as well as intracellular data. We look at respiration and growth profiling, because in mammalian cells there's a difference between respiration and growth. One of the main tools that my lab uses is constraints-based flux balance analysis. And within that, I will today allude on sampling of the flux space. Some, probably if we have time, touch upon synthetic lethality and positive epistasis. And of course, when you collect so much data, you always have to have data-driven analysis. And when you combine all these two, we will end up with understanding some of the mechanisms of resistance and sensitivity, try to predict chemosensitivity, and come up with a integrated paradigm for resistance, which all is under the realm of systems biology. Essentially understand the components, enumerate the components, understand their interactions, and eventually try to get to the basis of those emergent properties. So as I begin, let me draw your attention to this Pulitzer Prize-winning Cancer Whodunit by a very eminent clinician and scientist, Siddhartha Mukherjee. And one of the quotes he has in his book is, how can one capture genes that behave like ghosts, influencing cells from behind some dark curtain? So in my opinion, this quotation refers to, although we know that cancer is primarily a genetic disease, refers to the downstream products, to the different molecular hierarchies that exist in the cell that interact with each other and eventually make it very difficult to understand the emergent pathogenesis of cancer. Another quote, that cancer exploits the fundamental logic of evolution, just like we as a species are the ultimate product of Darwinian selection, essentially tells us that we have the right of way to use this method, the constraints-based modeling and analysis method, and the principle of optimality that flux balance analysis purports to understand the emergent properties of cancer. So we all know that cancer begins when a single cell begins to grow out of control. There are many hallmarks of cancer that have been delineated over the years, and they're very well summarized in this review. So what is very interesting here is that although cancer has been studied for the past century, metabolic hallmarks have emerged only in the last decade or so. And that's probably because the links between the different metabolic alterations and the non-metabolic alterations given in this inner circle, that research has come about only recently. So this figure here has these, I like to call them centripetal arrows moving outside. They show how the non-metabolic hallmarks of cancer, like avoidance of immune surveillance, tissue invasion, self-sufficiency in growth signals, etc., how they impinge on metabolism. And the centrifugal arrows, the ones that are pointing inwards, they essentially talk about the metabolic contribution to hallmark acquisition. So it is, why does cancer need a systems approach? That's because it's a multi-hit, multifactorial disease. And what I mean by multi-hit is that when changes occur in the cell, changes occur at every level of molecular hierarchy, right from the RNA to the protein to the genome and the metabolome. And it's multifactorial because it depends on different microenvironments that the cell is subjected to, the human is subjected to, including nutrient availability, the oxygen levels, chemotherapy, the environment, etc. And it's further complicated by two very unique features, that is metastasis and recurrence. So coming to one of our model systems, our model cancer that we study, that's glioblastoma. So glioblastoma is one of the primary brain tumors. It generally starts from cells of the brain. There's still a question whether it starts through stem cells. Is there a neural progenitor for that? It generally demonstrates infiltrative growth, so it's like mixing black and white sand together, and hence it's very difficult to diagnose this disease, and it makes it difficult to treat this disease. Some of the hallmarks of this tumor are increased metabolic demand, necrosis, and microvascular proliferation, and that's why it makes a very good model for us to study metabolism. The treatment, talking about the treatment of glioblastoma, the prognosis, as I said, is very poor. The treatment options are surgery, radiation, chemotherapy using temodar or temozolomide, which I will talk about in a little bit, Avastin. Tumor recurrence is very common. It happens within 15 months, so it leads to another round of therapy. There are very few experimental therapies that are available. CAR-T therapy has been tried but has been unsuccessful. Eventually, death occurs in a mean time of about 15.4 months. That's the prognosis. So new treatment options are desperately needed. Temozolomide is or temodar or Avastin is not Avastin, temozolomide or temodar is a DNA methylating drug. It methylates at the N7 and the N3 positions of guanine in the DNA, and that is how it prevents the cells from growing. The half-life of TMZ is about 1.9 hours. It is generally given as a prodrug, and so it forms these methyl diazonium ions, and the methyl groups from the diazonium ions move on to the DNA. The action of temozolomide can be counteracted by a gene called the MGMT gene, which is a methylguanine DNA methyltransferase. And what it does is it's a DNA repair enzyme, so it sort of reverts back what temozolomide does. This graph here is a Kaplan-Meier graph that shows that when you use temozolomide along with radiation, you increase the mean survival time. And so this drug combination and radiation combination was discovered for the first time in 2009. And by 2012, resistance began to develop. So it is very critical to understand this resistance and find new ways to circumvent them. So what is the model system that we use? So there's a very common cell line that you can buy off the shelf from the American Type Collection called the U87 cell line. It's a grade 4 cancer. It's a glioblastoma multiforme cancer. And we've had this cell line in our institute for a while. So we wanted to, as I told you earlier, I look at metabolism. So I wanted to look at experimental methods to extract metabolites and to quantitate them. So we started growing these U87 cells in flasks, in T flasks. And what we observed was, or what my student observed was, that these cells began to float on top. There were a lot of cells, when you grow them up in the numbers that you need for these experimental analysis, they come up. And she was very sure that these cells are dead, because in mammalian cell biology, if a cell comes up, it's dead. So it happened multiple times, and then we realized that it cannot be that these cells are dying all the time. So we started observing the morphology. We did other characteristics. We stained them. We looked at them under the fluorescence activated cell sorter. We added dyes. We looked at and when we did this cell sorting, we were able to separate out this fraction that floats and the fraction that remains adherent in the flask. And they were indeed different types of cells. They were separated using the hexed dye. As a chemical engineer, I like to do growth curves. I like to calculate doubling times and growth rates and lag times, etc. And so if you look at the graph on the left side, this is the parental U87 cell line, which grows at with a doubling time of about 36 hours. And if you separate the two cells out using FACS, and we will call the red cells, the neurospheres, or the cells that float on top, which have a spherical morphology, neurospheres, and U87Mg is the one that's what we have purchased from ATCC. That's the adherent cell line. We will always denote the U87 with a blue line, and all data for NSP will be denoted with a red line in the subsequent slides. So as you can see here, neurospheres have a doubling time that is much higher, so they grow much slower. Then the next question we asked was, are these floating cells responsible for drug resistance? Are they responsible for metastasis? So we were not able to check the metastasis hypothesis out, but definitely we were able to test the drug resistant or drug tolerance hypothesis out. And we see that the IC50 value, the half lethal concentration for temozolomide, for the neurosphere cell line was almost 40% higher, about 1,000 micromolar, as compared to that for U87Mg, which is about 745 molar. So the next question we asked was, are these stem cells? Because there's a lot of question about where these cells come from. And what we did was we added certain differentiation factors to these spherical cells and saw that indeed after 4 days we were able to see differentiation. So now this is all good. We've set up a model system. We want to understand what is this primary. We've established a primary phenotype. We know the drug response. We've seen the growth. Now we need to connect this to the genotype, because we want to understand the genotypic basis of this difference. And we can connect these to a lot of secondary phenotypes like respiration, metabolism, nutrient preferences, to understand selection pressure driven evolution. So how do we actually go about doing this? So as you can see, it's a very complex problem that is stated over here in a very simplified manner. So you need to use both experimental as well as computational methods for the same. So one of the computational methods that I alluded to earlier on was the constraints-based analysis and network reconstruction method. So I will now move on, switch gears and move on to the pedagogical part of the talk. Where we can create a model, we can create a metabolic network for studying glioblastoma and talk about why and how can we incorporate different constraints into this model. And eventually, I will talk about some of the results from our research. So in order to do this, you start with the primary annotation of the genome. And if you look at this toy example of DNA, it has 3 genes, the green, the red, and the orange. Now, these genes can have varied gene products. So assume that this green ORF, open reading frame, codes for a circular DNA, a green DNA, which codes for a regulatory protein that can bind to the DNA and either transcribe or repress it. Let's assume that this red open reading frame codes for a triangular protein, and the orange open reading frame codes for a square protein. And both of them can come together to form a complex, and they can catalyze a reaction, for example, an isomerization reaction that converts A to B. So this is essentially the primary annotation of a genome, right, or of genes. Now, what our lab likes to do is, our lab likes to represent these in terms of a mathematical formalism. And what we do is, it is quite simple. We look at what are the components that participate in reactions. That is the rows over here. So in the case of this DNA that transcribes a regulatory protein and represses a gene, you have the green circle, the purple DNA that it represses, and the DNA protein complex. For the protein-protein interaction, you have this triangular protein, the square protein, and the protein complex here. And the metabolites that are mentioned here in an isomerization reaction are A and B. So what's easiest for you to understand is to focus on this lower formalism. So here you have a vector. This vector represents this reaction, A going to B, and because A is consumed, it gets a value of negative, and since B is formed, it's positive. And you can use the same logic to understand the protein-protein interaction. So there's a combination of both these proteins together to form this protein complex in a ratio 1:1, and in here you have the DNA binding in a ratio of 1:1 again. So essentially, the stoichiometric elements form the elements of this matrix, and the sign essentially shows what direction the reaction is going in. So you can see that with just 3 open reading frames, we already have an 8x3 matrix, right? So imagine if you have to do this for the whole genome, you would have, you would have a very large matrix to work with. But what is the usefulness of a matrix is that it is then amenable to the tools of linear algebra, convex analysis, etc. So essentially, if you want to zoom into the glycolytic pathway in E. coli, if you look at the first reaction, the hexokinase reaction, this is the reaction hex 1, and this is the chemical reaction vector that defines this reaction. The only components that participate get elemental value, and the rest are denoted as 0. The direction of the reaction, the substrates and the reactants are understood by the sign of the elements in the matrix. So this is the outermost layer that anybody sees, that any mathematical modeler can see and can work on. But if you really want to understand biology, you need to, as I mentioned in the first or the second slide where we saw the interactions of all the molecular hierarchies, you need to incorporate you need to incorporate what is the genetics behind this metabolic reaction. So these models include this information. So if you look on the right-hand panel, herein you see a monogenic relationship, one gene, one protein catalyzing one reaction. Herein you see an isozyme representation. So you have one gene, one protein, another gene, another protein, or an isozyme, and both of them catalyze the same reaction. Below here is depicted like a protein complex kind of association. So all these are represented in the model as Boolean representations. So you have an outermost layer, which is the matrix, but behind this matrix, you have associations for the gene, the transcript, the protein, and eventually the reactions which are depicted here, you can put in information about the metabolites. And this way you can tie up the biochemistry, the genetics, and the mathematics together. And you have different modules that you can create, so you can study each one of these at a time, or you can study them all together. This is just a example network that I'm showing here of Recon-1, one of the first human metabolic networks created. So by putting together all these modules, you essentially can represent different organs. You can represent different pathways, different genes, transcripts, proteins, etc. So you can see the extent to which the first model for human metabolism represented physiology, that is with 98 pathways that coded for 3,311 reactions, 3,311 reactions, each using 2,700 approximately metabolites, and these were coded by 1,496 genes, which resulted in 1,905 transcripts. So what I'm trying to show here is the extent, the complexity of these networks, the complexity of the matrix, and the fact that when you have such a large matrix, you as computational biologists can explore them further. So I've been talking about this matrix and the matrix construction from biology, right? So where does the math come in? So once you have this matrix, the S matrix is actually, if you remember your high school algebra, every matrix A, when you multiply it by vector X, which produces a vector B, is essentially a linear transformation that maps your vector X to your vector B. So here in biochemical reaction networks, this S matrix actually acts like a linear transformation between these reaction rates that are given in vector V and the time derivative of the concentrations, this dx by dt. And these transformation, this linear transformation is essentially defined by this mass balance equation. All of you must have, whoever is familiar with flux balance analysis must have seen dx by dt equal to x dot V in multiple textbooks and multiple talks and so on and so forth. But this is why this transformation is so useful, because it represents S as a linear transformation. And if you go back to your linear algebra knowledge from high school, if you remember, a matrix can be represented as there are four fundamental spaces of a matrix. And essentially, the row space and the null space fall in the solution, the flux solution space of a biochemical reaction network. And the column and space and the left null space form a part of the concentration solution space of the network. Now, what has been explored till now in this field of constraint-based analysis is generally the flux solution space. So it is the null space that gives you the solution to understand your biochemical reaction network. So what is still open to a lot of research and analysis is the left null space. And this is the left null space is the one which shows you information about the concentration. And if you look at the way experiments and experimental measurements are done, it's concentration that we measure. So for people who are interested in metabolic networks, metabolic network analysis, constraint-based analysis, and think that everything is already done by researchers in the world, this is one space where you all can work in, looking at the mathematical analysis of the left null space. So in yesterday's talks, I think flux balance analysis was alluded to, but rush through, so let me just give you a quick overview of what it is. It's essentially you have the reactions in the cell, you have the reaction stoichiometry defined, you define this transformation S dot V is dC by dt. At steady state, where since the metabolite turnover rates are so fast, you can equate that to 0, which is also a property of a linear transformation. So, so essentially you end up with an underdetermined system with fewer equations than unknowns, and therefore you have infinite solutions, and therefore you need to call in the idea of linear optimization, which is subject to linear equality constraints, and you end up with one solution which is relevant, either the highest or lowest value of that objective function. And this is again very suitable to apply in biology, because we all believe in, or most of us believe in Darwinism, and we think that it's survival of the fittest, right? So you will always, an organism or a cell will always move towards optimality in terms of its fitness or growth. So in interest of time, I will skip this constraints-based philosophy slide. Essentially, it says that when you have a space of everything that some bacteria or cell can do, you apply constraints, and then whatever remains, however improbable, that is the solution. That is what the cell is doing. There are many constraints-based methods that have been developed till date. And generally, this philosophy can be easily applied to biology, modern biology, because of all the measurement techniques that have been developed over the last 2 decades or so. So with all the omics data that's coming up, you can integrate this omics data into these type of models, and you can probe them to understand you can compute the phenotype of the cell. So what did we do for glioblastoma? We did the same thing. We looked at what were the growth limiting metabolites for the 2 cell lines. We found a differential profile for them. We looked at the respiration on different compounds. As you can see, there are different profiles for the blue and red lines, and that means that these compounds are used differentially. We typed the genome. We wanted to understand what are the genome changes. So we looked at the whole genome sequencing of both these cell types and found that they were riddled with mutations, almost 30,000 on an average SNPs in both these cells. We thought we might be able to find differences, non-overlapping mutations, but we saw that more than 11,000 were overlapping, and there were only a few that were unique. And 7%, what was interesting to us was 7% of the mutations were metabolic in nature, and so, and within that itself, there was a distribution of common and unique between the 2 cell types. So using all this information, including the metabolic, the exometabolome profiles to develop uptake rate constraints, we used flux sampling, and flux sampling essentially, as written here, gives you the range of feasible flux solutions and also the probability of their occurrence. And you can span the entire solution space, and this is what I really like about it, and you can generate these feasible solutions without what is called an observer bias. Because you're spanning that whole space. And what we identified was these were a few set of pathways. They look like a lot, but if you track back to that slide where I showed human metabolism reconstruction, there are 98 pathways. So 5 out of 98 is not really a lot. And we saw that there was a difference in glycolysis, in the TCA cycle, the malate aspartate shunt, and the folate pathway, and so on and so forth. And based on this differential programming between the 2 cell types, we did a chemosensitive profile to see whether we could see any differences in the way these cell types respond to drug molecules. And if you can see here, there are a bunch that are similar and there are a bunch that are different. I'll just show you one example here. So we did a screen first. The previous slide was data from a screen. And herein we took one of the compounds, rotinone, which is an electron transport chain inhibitor. And we did a drug dose response curve for them. And if you look at the left and the right-hand side, and the colors have inverted for some reason, this should be blue and this should be red. you can see that the IC50 for U87 is 1.8 micromolar, whereas that for NSP or the neurospheres is 5 nanomolar. So there's a 3 order of magnitude difference in the amount of drug that is required to kill these otherwise resistant cells. If you remember the temozolomide drug dose response curve, it was 40%. The IC50 was 40% higher for NSP as compared to U87. So in summary, such an integrative analysis of data in the context of a constraints-based human network model will identify different reprogramming. We've identified a programmable switch in those 5 pathways that I mentioned using Monte Carlo sampling. I haven't alluded to the ETC function or the cholesterol metabolism or any of the synthetic lethality work in interest of time or because of lack of time. But anybody who wants to talk to me about that, please feel free to talk to me later. And essentially, what is the future direction? That such integrative systems level approaches indeed work. They are critical to unraveling all the tacit connections between epigenetics, metabolism, transcription, and the genome. And we are currently talking to some clinics, and we are trying to see whether we can establish a similar difference using patient-derived cell lines from glioblastoma. But because it's glioblastoma, it's really hard, because once you do the surgery, you need the cells right away. So no work can be done without acknowledging funding, without funding. So I thank all my funding sources. The work that I showed was mainly done by Rupa, an ex-PhD student who's now at ISB in Seattle, and Avinash, who was a joint PhD student with me, and he's now at Francis Crick Research Institute in London. This work is being continued by a very talented postdoc, Vijendra, in my lab, and another graduate student, Madhuri. And with that, I'll leave you with the picture of my group, and thank you. And thanks, and hope I have some time to take questions.
Thanks a lot, Anu. Very nice talk. So do we have some questions, please? Right, I'll start with one. So when you said that the DNA mismatch repair enzyme, the DNA methyltransferase, I'm so sorry, is the one which is actually taking care of the epigenetic modifications that TMZ brings in. Do you see any difference in the neurospheres compared to the ones that are adherent to the...
in the mutational profile? Yes. No, we didn't.
So you didn't pick up anything?
Yeah, so this is truly whatever changes we are seeing is due to metabolism, and it is not related genetic to that genetic. Yeah.
Nice. Yes, please.
So you made context-specific models for each of these?
Yes. So we've made with the...
integrating the transcriptome.
Not transcriptome based, but uptake rate based. So exometabolome based. And we also use our genome data, wherein we change the bounds of our reactions based on what type of mutations we see in these genes. So if we have a non-synonymous versus synonymous mutation, we have a different formula that we apply to restrict changes. Because although synonymous mutations are not supposed to change the protein function, what we have looked, when we look up in literature, they do have some transcriptional effects.
Could change,
yeah,
yes.
So the levels could change. So we constrain them by a certain algorithm, and if it is a non-synonymous change, we reduce them quite a bit. Yeah, so we constrain our flux solution space that we are searching for our solutions in.
Thanks for a very nice talk as always.
Thank you.
Hi, nice talk. This is the rotenone sensitive and tolerant cells, right? So the one which is tolerant, did you, try using oligomycin for those cells?
Yeah, so we are currently working on that. So we've looked at all, so since we identified, so I didn't have time to show, but we've looked at all the mutations in the electron transport chain, both in the mitochondria, the genes that are coded for by the mitochondrial genome, and also the somatic genome. And there are mutations in some of them that are different. There are, we've looked at different inhibitors of different complexes. I can't recollect as of now. Oligomycin is an inhibitor of complex 4, right?
So basically so the reason I'm asking this is, when you are having something which is tolerant to rotenone, there are many ways you can explain it. One is, of course, rotenone is not able to bind and execute because of mutations. But other is, if cell is not relying on ETC for its energy needs, right? And a lot of these transformed cells actually undergo just aerobic glycolysis. And they use ATP generated by glycolysis and expend that energy using F0, F1 ATPase to, ATP to ADP conversion and maintain mitochondrial potential. So those cells will be more sensitive to oligomycin than rotenone because the ETC is not happening that efficiently in those cells.
I see. So what we see is that between these two cells, the rotenone sensitive cells are the ones that prefer using glutamine, and the ones that are more tolerant, they prefer using glycolysis. They use glucose, and that pathway is more upregulated as compared to the other cell type. And we are also pursuing another hypothesis where we feel that there is a dissection that happens in the mitochondria between proton pumping and ATP formation, because I didn't have time to talk about all the mutations.
That's what I'm saying.
Yeah, so ATP synthase is mutated in one of these cell types and it's not in the other. And there's another gene that can allow for recycling of ATP. There are differences in those genes as well. So we have a feeling that eventually it's the electron transport chain function that is differential in both these cell types. And because we see that when we use these different inhibitors, the inhibition is very different between these two cell types. But those experiments are still in progress, so maybe the next time, and maybe we can discuss the results sometime.
I have a general question, if you don't mind it. Biological systems are generally characterized by nonlinearity, right? Brain behavior, even gene regulation. But metabolic systems seem to be characterized by a linearity. Is that impression correct? And if yes, why do you think that's the case?
That's a difficult question to answer because anything I say can be right or wrong. So I don't think metabolic, I think I've lost my water bottle. So I don't think metabolic systems or metabolism is linear in the true sense, but I think in the way we are looking at it, I think we have sort of simplified it we've simplified it in such a way that it looks more linear, and so we are able to do these kind of analysis. Because if you ideally look at it, I think there was one talk yesterday by Neera where he showed how you integrate your signaling networks with your metabolic networks. So essentially, I mean, you do not know what are the other, so for example, what are the other signal pathways that are being utilized by this cell. We completely ignore that when we are currently looking at this. It's not an integrated model. So it's just an independent model of metabolism. It's sure it's a little large, it represents what happens in the cell, but still it's isolated. But I think when we start putting in the different layers of hierarchy, when we actually look at when our models represent the interaction between signaling, between metabolism, I think that's when we will see a lot of non-linearity. Because for example, a mammalian cell is not cell autonomous for taking up any of these nutrients that I mentioned. So if the tyrosine kinases, if the AKT, the PI3K pathway doesn't get turned on, even the cell is dying, it's not going to take up the glucose that is there. So, I don't think it's so it's not really linear, but it's a simplification that we are looking at as an isolated metabolic network. So sure, these represent the human cell in silico, but to understand the physiology, you need to add in more layers.